I want to welcome and thank you to everyone for joining us today. A reminder that this is a Zoom webinar, not a Zoom meeting, so you can hear and see us, but we cannot see or hear you. However, we will be taking your questions at the end of the presentation. You should see a Q&A icon. So go ahead at any time and type in your questions, and then we'll try to address as many as we can at the end of the presentation. We have a lot of material to cover, so we're going to be moving things at a rather fast clip. And we'll start with introductions. I'm joined here today by three of my colleagues. Dr. Randy Carpenter. He's a physician by training, spent a number of years in academic medicine, and then transitioned to pharmaceutical companies. He's worked for large companies, medium companies, small companies, biotech companies, including a startup, which he co-founded and was CEO of Seaside Therapeutics, that was developing drugs for Fragile X. We're also joined by Dr. Jana Van Heen. Jana is a molecular geneticist. She heads up a lot of our clinical research efforts, and you'll be hearing from her later. Between Dr. Carpenter and Dr. Van Heen, they have decades of experience in business development, drug development, clinical trial experience, all of things that are incredibly important and relevant to what we're trying to achieve at RSRT. And then finally, we're joined by Tim Freeman. He's our Chief Development Officer and fellow Rett Syndrome dad. I'm sure many of you know him already. So despite the incredibly serious and debilitating nature of Rett syndrome, the disorder does offer us three very important key advantages. Number one, the root cause is known. Unlike disorders like autism, schizophrenia, Alzheimer's, Parkinson's, depression, MS, for which the genetic scenario is very complicated, for Rett syndrome, it's much simpler. We're a monogenic disease. We know our gene, causative gene. The disorder is not also neurodegenerative. Brain cells don't die. They're all there. They're all in the right place. And then finally, we know from work that was done by Adrian Bird that restoring levels of the MECP2 protein dramatically reverses symptoms in mice. And so because of these three facts, we believe that the path most likely to deliver dramatic recovery is a genetic solution. So in other words, fixing the root cause of the disease. And so after many years of funding critical basic science, in 2017, RSRT launched Roadmap to a Cure. It was a 3-year, $33 million research plan. And the goal was to identify and to launch or expand therapeutic programs that target the root cause of the disease. And I think anyone that looks at this graphic can see that there was a tremendous amount of activity between, during the 3-year timeline of Roadmap. And without getting into any specifics, I think the take-home message here is that science is rapidly advancing and that we are hyper-vigilant to emerging technologies that can be leveraged for Rett. And as these technologies presented themselves, our programs expanded. Roadmap generated substantive results. With the amazing support of the Rett syndrome community, we successfully raised the $33 million necessary dollars that were needed, and we were able, therefore, to expand and launch 15 programs in 6 strategic areas. I think most importantly, at the beginning of Roadmap, there were 0 companies that were focused on MECP2 therapeutics, and now there are 6 with more to come. There were a lot more results and accomplishments that Roadmap achieved, but rather than take your time now to read through these, I would invite you to visit our website, click on research, and then click on accomplishments, and you'll be able to see in more detail the types of results that Roadmap generated. So in recent years, RSRT has transitioned from simply an organization that funds research projects into an organization that incubates research programs for the explicit purpose of transitioning these programs into biopharma companies. And this is a really important point because while academics are the ones that make discoveries, typically, they are not drug developers. Drugs are developed in biopharma companies. And so being able to make that transition from academia to biopharma is incredibly important. And to encourage that transition, a couple of things need to be in place. First of all, we need a strong portfolio of foundational work. We need to understand, for example, as much as we possibly can about our gene. And in our case, the MECP2 consortium was instrumental in making that happen. You have to have freely available animal models. You have to have patient tissue. You have to have molecular tools and clinical research efforts, for example, like our biomarker initiative. All of these have to be in place to attract biopharma interest. And I think Rett syndrome now is in a really good place with regards to these foundational resources, thanks to the millions of dollars that have been invested in this type of work. I think the other thing that has to happen is that you need to be thinking about business development right alongside the science. And that means paying attention to things like intellectual property, to licenses, to options. And in this regard, we're very fortunate to have Dr. Carpenter on our team, as he has a lot of experience with business development. I think a good example of how RSRT has transitioned into the role of incubator is in the gene replacement space. So we started funding ad hoc gene therapy projects over 10 years ago. We funded Gail Mandel, Adrian Bird, Stuart Cobb, Ron Crystal, Brian Kaspar. And then there was increased focus as data started to be generated, and it morphed into the Gene Therapy Consortium, which then generated the data which made it possible to transition into company programs at Novartis and Taysha. And I think the important thing here is that the dollars, the donations that were made to RSRT that enabled us to fund the gene therapy projects and the Gene Therapy Consortium have now been leveraged many-fold because companies like Novartis and Taysha now have hundreds of millions of dollars that they can use to support their Rett programs. So now this takes us to Cure 360, which is our transition, our next step after Roadmap to a Cure. And we have increased focus now on these 6 strategies that you see here. And by the way, please do visit our website. You will find this graphic that's interactive. You'll be able to click on any of these and learn more about it. And also for time's sake, we're not going to be covering today these resources, clinical and scientific resources. But again, you'll be able to go to the website and to read more about each of those efforts. So you might be asking yourself, well, why 6? Where did the 6 strategies come from? Well, first of all, we are funding all of the strategies that are available today with today's technology that allows us to boost levels of MECP2. There isn't something out there that we're not pursuing. So that's how we came up with these 6. It's all of the available technologies. I think second of all, we're pursuing all 6 because we don't know which one will work the best. And also because it's the nature of science and business that perhaps not all 6 of these will move forward. And so I think it's important to have as many shots on goal. And we're going to take you through each of these. Actually, Dr. Van Heen is going to take you through each of these technologies starting right now.
Great. Thanks, Monica. So over the next several slides, I'll briefly describe the status of our curative therapies, which Monica has mentioned are all MECP2 focused and aim to correct either mutations in the DNA, in the RNA, or to deliver healthy protein. So let's start with gene therapy, which is the closest to the clinic. All right, so right now there are 900 open INDs at FDA, which means there are 900 different gene therapies in development for use in the United States. And the FDA will monitor all of these programs, and then they use this collective knowledge to maximize the safety of all gene therapy trials. So what's really important about this point is that FDA knows more about gene therapy than any single company or academic lab alone, which is great. And so even though FDA has approved a number of gene therapies so far, only 2 of them would fall in the same category as Rett, which is to say a therapy that's dosed inside the human body to cure a genetic disorder. And that's great news because it means that the path to approval is known and safety and efficacy requirements to achieve approval has been defined for companies. All right, so the first trial anticipated to come to the clinic is the Avexis, now Novartis Gene Therapies trial, utilizing AVXS-201. It's been renamed to OAV201. And Novartis Gene Therapies is committed to moving this program forward as they indicated in their community statement, which was released in August. And it stated very clearly that they're working with FDA to get to the clinic as quickly as possible. And what's important to note about this trial and gene therapy trials in general is that they're not going to follow a traditional phase 1, phase 2, phase 3 approval path that other types of treatments typically follow. More like oncology drugs, gene therapy is not tested in healthy volunteers first. It will go right into patients. And the doses that are used must have a therapeutic benefit. So that means that if results are really dramatic, it's possible that the approval path may be significantly shorter than standard therapies, and it's possible that they could potentially be achieved in one study. So we know the question on everyone's mind is when will this gene therapy become available? And that's a really difficult question to answer because this field is such a rapidly changing landscape that there's really no way to predict that right now. But RSRT is supporting Novartis Gene Therapies in every way we can. We are sharing our data on biomarkers, on outcome measures, and we're working with them on the registry as well. And so other gene therapies in development for Rett are Taysha Gene Therapies, as Monica mentioned, another technology that was spawned from RSRT's Gene Therapy Consortium. And Taysha has raised more than $200 million just in the past 5 months, with Rett at the top of their pipeline alongside another indication, which is great news. And there are 2 additional programs we're funding in development in academia as well. All right, so what are the goals of the gene therapy? So gene therapy experiments show greater efficacy in the Rett mouse than any therapeutic tested to date. And mouse studies are really important because they tell you how much correction is needed to improve function and extend lifespan. And they give you really important information on safety. But mice alone are not enough before you go into humans. You need to have animal models with larger brains. And so animal models with larger brains, like non-human primates, give you really great information about safety as well. But because their brains are much larger than those of mice, they can also answer important questions about distribution of gene therapy. And in Rett, you really want to see as wide a distribution in the brain as possible.
I just want to jump in and make one point, and that's the non-human primate experiments that are done in gene replacement trials are on normal monkeys, not Rett non-human primates. So they're not disease models. So you're doing those experiments, as Dr. Van Heen explained, for safety and distribution of the gene therapy product, but not to look at symptom improvement.
That's a great point. Thanks for making that, Monica. All right, so let's move on to our second curative approach, which is gene editing. So this technology aims to change one letter, one DNA base in the DNA code and correct the mutation in MECP2 back to normal. And this is a mutation-specific approach. Beam, the company pursuing this technology for Rett, was founded by 3 scientific leaders who have made really amazing breakthrough discoveries that have enabled base editing. And Beam leverages decades of this basic science research, millions of dollars in NIH funding, And as a company, they've raised over $400 million. And our investment in Beam allows them to focus these resources specifically on Rett. So we're able to move Rett to the top of the list. Okay. Next slide, Monica.
You want to say anything about mutation direct?
Yes, so this is a mutation-directed approach. So each mutation would require its own strategy. And right now there are 3 ongoing clinical trials using CRISPR. And what's interesting to note is just today, the Nobel Prize was given to the discoverers of CRISPR. So this is very exciting news. Okay, and then our other DNA editing strategy is gene exon editing, where rather than editing a single base in the DNA, you can replace chunks of DNA. And the advantage of this particular strategy is that if you can replace a stretch of DNA that's prone to mutations, you end up correcting a large number of different mutations with the same corrective therapeutic. So it's one therapeutic for multiple mutations. And this program is looking to generate a therapeutic that can correct 97% of mutations. The scientists working on this project are trying to generate proof of concept that this approach can work. And if they're successful, will provide the basis for investors such as VCs, public investors, or pharma to fund studies that allow this to move into pharma. And in early drug development, there's a situation where the basic science needs to move from academia to development to get picked up by a company, and that is called the Valley of Death. And what we'd like to do for this strategy and all of those that are still in academia is that we'd like to generate enough data that a company can pick this potential therapeutic up and develop it, that it's been de-risked to a level that there's confidence that taking this therapeutic forward is worth doing. And right now, this program is still in an early stage. All right, so the third strategy, MECP2 reactivation, this focuses on the fact that females have 2 X chromosomes in every cell, and that a normal copy of MECP2 is present, even in cells that are expressing the mutation. By waking up this silent normal copy, you could, in theory, cure every mutation. And recent scientific breakthroughs have occurred in this exciting area. This technology, however, is the only one that we'll discuss today that doesn't apply to boys, and that's because boys only have 1 X chromosome. To date, RSRT has funded 2 approaches to MECP2 reactivation. Thank you, small molecules or drugs, as well as biologic approaches, which use an enzyme like CRISPR and DNA editing. And these programs are advancing, and there is interest from pharma in pursuing these approaches, which is really exciting.
I'm going to jump in for a sec too, and Dr. Van Heen mentioned that the MECP2 reactivation wouldn't apply to boys with Rett syndrome. But I think it's also important to mention that all of the other research that we are funding, all of the other strategies, with the exception of MECP2 reactivation, does apply to boys with Rett as well.
Great. All right, and so then our fourth strategy, RNA editing, focuses on correcting the copy of the DNA and not the DNA itself. And this corrects mutations in mutated RNA, leaving the normal MECP2 RNA alone. And there are 2 companies pursuing RNA editing, Vico Therapeutics and Shape Therapeutics. And Vico Therapeutics was founded with technology that came directly from our MECP2 consortium, and that technology has formed the basis for their Rett program. And to date, they've raised $31 million. Shape Therapeutics, whom we are working with to support and expedite their existing Rett program, has raised $36 million. Oh, and this is also mutation directed, so it will apply only to certain mutations can be corrected with these technologies. All right, and then our fifth strategy now is another RNA editing methodology called trans-splicing. And this technology changes an entire portion of the RNA rather than just making a single change in the RNA. And similar to DNA exon editing, it can address multiple mutations that occur in a similar region of MECP2. And this is a therapeutic that could correct mutations in 97% of cases.
And let me just make a point about that 97%. So 97% of all mutations are in exons 3 and 4 of MECP2. You can think about exons like chapters in a book. So MECP2 has 4 chapters, 97% of the mutations are in chapters 3 and 4. 3% of the mutations are in chapter 1. So one therapeutic would address 97%. We could, and then there could be the development of a second therapeutic that could address the other 3%. So it's not that they couldn't be addressed, it's just that they wouldn't be addressed with this first therapeutic.
Exactly. All right, and so then our 6th and final curative strategy that we'll cover today is just straight replacement of normal MECP2 protein. And to date, it's been very difficult to deliver proteins to the brain since they can't easily pass the blood-brain barrier. However, there have been some really cool breakthroughs in this area, and if this is successful for Rett, it could form the basis of a new platform-based company where Rett could be a lead indication if proof of concept work is successful. And so this technology is really focusing on getting protein to pass into the brain from the bloodstream. And this is a really cool breakthrough that has not been available for Rett or for any other type of neurological disorder. So this could be really groundbreaking if the proof of concept work is successful, and we're really looking forward to finding out if that's true.
Probably worth mentioning that this is one strategy that would require ongoing dosing, not a one and done approach. And while the one and done certainly has advantages, there are also advantages to repeated dosing, because then you can titrate the dose as necessary.
Right, so if you needed a little more protein to get better function, that would be possible, or if you saw some side effects that were worrisome, you could reduce the dose to eliminate those. So there's a lot of great possibilities here.
Okay, so this is a MECP2 therapeutic drug development pipeline, and it's on our website. I know it's a lot to take in, so don't feel that you have to memorize this or take a screenshot. It's on our website right now. And I'll draw your attention to a couple of things. So on the vertical, you'll see the 6 strategies, therapeutic strategies with each of the programs that are taking place in each of those strategies. And across the top, horizontally, are the various phases of development, therapeutic development. The orange are the academic programs, and the ones that are magenta are the biopharma. The circle is where we are today, and the arrow is where we expect to be in 2024. And of course, we have shared this graphic with all of the companies that we're working with and all of the various programs, and this has their blessing. So, but one thing I would stress is that this is going to be dynamic. Expect change. Some things will move faster than expected. Some will move slower than expected. Some of these may drop off. There will be additions. So this is a snapshot in time, and we will be updating this often, I suspect. So those of you that know me know that I've been doing this type of work for a long time. My daughter is going to be 24 on Saturday. I've been working with the scientific and clinical community for 22 years. And the way that RSRT operates today is actually very different than we did even several years ago, let alone 10 or 15 years ago when I was working at my first organization that I helped to start. And I thought it might be helpful to go through kind of some of our core philosophies on how we approach funding research. And first of all, I'm sure it's abundantly clear by now that we are, what I like to say, MECP2 centric. We have limited funding. limited time, limited staff, and we really want to focus on efforts on what we think will deliver the most improvement for our children. And we think that those will be genetic solutions. And so we are MECP2 centric. We spend a lot of time, all of us do, on vigilance for new technologies. That involves, staying up to date on who's doing what and jumping on those technologies whenever we think that they might be relevant for Rett syndrome. And fortunately, that happens often because science is moving at such a rapid pace. We don't do requests for proposals, meaning we don't advertise to the research community and say, submit proposals to us. Anyone can submit a proposal to us at any time. But rather than do broad RFPs, which kind of require us to sit back and hope that somebody submits something that's exciting, we do the reverse. We identify the strategies we're interested in, and then we recruit the top people in each of those fields and invite them and encourage them to submit proposals to us. We provide sufficient funding and resources to them so that they can make progress, which will then hopefully will facilitate biopharma investment. Whenever possible, we avoid having our funded researchers working in siloed environments, but rather we encourage consortiums or collaborations. They're not all formal consortiums like the MECP2 consortium. Sometimes they're more informal that kind of happen after the fact, for example, like our RNA editing consortium. We focus on creating programs rather than funding ad hoc projects. And as I mentioned in the introductions, we have internal staff with drug development experience, clinical trial experience, and business development experience, which is crucial because at the end of the day, we're trying to develop a therapeutic. And then we have ongoing frequent interactions with scientists, with academics, with biopharma, and more with venture capital investors as well. So before we transition to Tim, the question that is on everybody's mind, including myself, of course, and Tim, and every parent, grandparent, anyone that loves an individual with Rett syndrome is, what will a cure look like for my child? And we can't answer that right now. We don't know until clinical trials for these therapeutics that target the root cause of the disease begin. We really don't know what that cure will look like. I think common sense tells us that early intervention will always be better. Intervening in a 1-year-old, if possible, or 2-year-old will certainly be better than intervening in a 30-year-old. I think we all kind of inherently know that. And I certainly don't expect a cure for my 24-year-old daughter, like a never even happened cure. But I do have an expectation that with a therapeutic that addresses the root cause of the disease, that she may be able to have dramatic improvement for the first time in her life. And I think there's a lot of area between having Rett syndrome and having a full cure like it never even happened. And so our mission and our hope is that we will have a full cure. How that will look like for children and adults of different ages remains to be seen. But I, for one, want that opportunity to see how much improvement we can have. Okay, I'm gonna turn things over to Tim.
Hi, everyone. Thank you so much for joining us today.
One thing, I just want to mention, we are going to do Q&A at the end. So once Tim is finished, we'll handle your questions.
Thanks, Monica. So I'm Tim Freeman. I know so many of you. I'm RSRT's Chief Development Officer. I'm also the very proud father to an 11-year-old daughter with Rett syndrome, as Monica said. My role at RSRT is to encourage and support families in fundraising for this research. And all this promising research simply cannot happen without funding. So I thought it would be helpful if I give an overview of how philanthropy works and also where our funding comes from at RSRT. And I get excited about this stuff. I'm excited to give this presentation because I often find that families are surprised in a good way and in an empowering way by some of this information. So this first slide shows all donations in the US to all causes last year, 2019. So health, education, religion, a total of $450 billion was given away last year. Almost 80% of that comes from individual people or family donors. 17% comes from foundations, and just 4% comes from companies. Often I find people are quite surprised that corporations are such a modest piece of this pie. So the next slide, this is sources of funds to RSRT over the last 3 years. And you'll see it's almost identical to the previous pie chart. In fact, individuals play an even bigger role at RSRT. Some of these individuals here are Rett families, but many are the networks of Rett families, their friends, their contacts who give when they're asked through events or through crowdfunding. So corporations and foundations, again, are a pretty small piece of the pie. So the point behind this is that it's all about individual people. They're the key to giving, not only to Rett research, but to all causes. So the next slide shows that virtually every dollar contributed to RSRT can be traced back to a family affected by Rett syndrome. Or another way to say that is we get no random donations. Interestingly, even when we get national press coverage or a segment on TV, it doesn't typically generate funding. The only way that funds come to us is when Rett families take action and raise those funds from their networks.
Tim, I might just add that that's not unique to Rett syndrome.
Yeah, true.
Common, scenario with lots of other disease organizations as well.
Yes, it is. I've talked to my counterpart at cystic fibrosis and exactly the same. So this slide, you might say to me now, okay, that's great, Tim. How do we take action? What do we do? How do we raise those funds? And here are the ways that we know from successfully completing Roadmap to a Cure are the most effective ways of raising funds. So 45% of our funding over the last 3 years came from events. And when I say events, I want to be clear that I don't just mean big galas. I mean backyard barbecues and happy hours and cocktails in a home. And yes, also big galas. So every one of those events was started by a Rett family. They invited their networks to support, and their networks happily contributed. 40% of our funding comes from what we call major gifts. Those are individuals and families who make a donation of $10,000 or more. 10% comes from crowdfunding, and most of that crowdfunding happens on our platform, which is rettgive.org, and through Facebook campaigns. And this is a very easy and effective way to raise funds. And given the pandemic and the impact it's had on in-person events, I suspect that more and more families will start Rett Give campaigns, and that 10% will very likely jump up in the months and years ahead. So if you do want to start a Rett Give campaign, email me, call me, and I'll walk you through it. It really could not be easier. The other, if you go back to that other slide, Monica, the other on this chart is just when a family has a connection to someone who has a foundation or someone who's especially philanthropic. In those cases, I'll work with each family case by case on an approach that is comfortable for them. And it's important to note that this really needs to be a personal connection that a family has. There are a lot of big foundations out there that have no personal connection to Rett syndrome. I've written letters to many of them. And unless there's that personal connection to a Rett family, they're not likely to fund this research. But if you do have a connection to someone who's philanthropic, someone who's a potential big supporter, it's definitely worth giving me a call and brainstorming with me about it. So the next slide, over the past 3 years, we've really been very, very fortunate. We've had more than 1,600 families fundraising for us and with us. Many of you are on the call now, and for me to say thank you to those families, it always feels like a big understatement to me, given how much we all care about our outcome here. But I will take a second to thank you for your energy, your dedication, and for being behind us so strongly. You are part of an extraordinary team of families who raised that $33 million over 3 years using the methods that I just described. So these families averaged raising $20,000 each. That's their power. That's all of your power to affect change, to make this research happen. And I ask you to really imagine if we doubled the number of families that fundraise for us. It would be quite remarkable. And the bottom line here is that the more funding we have, the faster and more effectively we can push this research ahead towards what we all want. So a few families have said to me, I don't want to fundraise in the COVID environment. And this slide is a poll of a random group that was asked how the pandemic was going to change their giving over the next year. And you can see the majority of people plan to give the same as they've always given. A quarter of them plan to give more, and just a very few, about 7%, plan to give less. So yes, absolutely, we have to be especially thoughtful in the fundraising we all do now. If you're reaching out to anyone who has been directly affected by the pandemic, but the vast majority of people are as eager and pleased as ever to donate to our cause. My last slide. So most people are philanthropic. They want to support a good cause, but they'll only do it when they're asked by one of the methods that I've mentioned here. If they're not asked, they're going to give their money somewhere else. And the way I see it, I'd much rather have it come to Rett Research. So most of you probably know already that we spend an average of 95% of our funding on research at RSRT. That's an almost unheard of overhead rate for a nonprofit. We're parents, Monica and I, so to us, any other way would simply not be acceptable. Maximum dollars have to go to changing our kids' lives, to the research. Fundraising can be fun. You interact with lots of people, you help them feel that they're doing something that's important, that matters. I find it empowering to know that you can do this, you can make a real difference. And finally, the dollars we raise really now have their biggest impact ever because we're leveraging all that was achieved during Roadmap to a Cure. So if you're not already involved with us, if you want to get involved, please contact me. I love hearing from families, not just about fundraising, but about your children, about life with Rett syndrome. I am here. Call or email me anytime. Thank you.
And one comment, it doesn't just have to be families in the US. Families outside of the US fundraise for us as well. And Rett Give crowdfunding is a great way to do that.
Yep.
Okay, I'm going to stop sharing so that and take a look at the questions that have come in. Okay, first question from Bjorn. Is there a difference between classic Rett and the milder version Zippela in potential treatment? The Zippela variant is a term that's used in Europe, not used so much here in the States, but it basically means preserved speech. And it's a small subset of kids with Rett syndrome that have some preserved speech. Is there it's a milder form. Let's put it that way. I don't know, Jan and Randy have anything more to add to that?
I mean, I would say as long as there's a mutation in MECP2 and that can be targeted, there's no reason that any therapeutic wouldn't be successful in that case as well.
Yeah, absolutely. Okay, Liz asks, what's an IND? Jan and Randy, go
Okay,
ahead.
great. So IND stands for Investigational New Drug Application, and it is it's how companies apply to the FDA to be able to start clinical trials in humans. And you have to have a package of safety data, of animal information. You have to have a sense of, the highest dose you can go to, your minimum dose that you can use. And so FDA evaluates all of this data from animals and data generated in the lab, and they look at your plan for how you want to treat humans, and then they approve that. And once you have an open IND, which means the FDA has granted you permission to start clinical trials, then companies can actually start putting their therapy into human patients in the form of clinical trials.
Great.
And I would just add that an IND is typically, the key issue is safety. Is it safe to take this compound into humans? And that's particularly for adults that can make their own informed consent. When you have individuals that are not capable of providing informed consent, they're considered vulnerable populations. So like children or children with Rett that can't communicate. And you also have to have the potential for the drug to be therapeutic. So there's two standards for Rett. It has to be safe, but we also have to believe that it will provide a benefit. So it's even stricter than a typical investigational new drug would be for a therapeutic that would be given to an adult who can make their own decision about whether or not to participate in a trial. So you can feel pretty comfortable that the FDA has deemed this to be safe and potentially effective if it advances into these treatments.
Great. Thank you, Randy. So there's a couple of people that are asking specifically about deletion. So would gene therapy or editing help Rett patients who have a deletion that is causing their Rett syndrome? So gene replacement therapy, MECP2 reactivation, RNA trans-splicing, and gene exonic editing are kind of generic treatments for anyone that has an MECP2 mutation. The ones that wouldn't work for deletions are the RNA editing and gene base editing, because those are mutation specific, and you need a particular therapeutic for every mutation. Okay, I hope, and if I'm not answering your question to the degree that you would like, please feel free to, submit another question. Okay, George says, you're explaining cures for mutation. Oh, please, okay, George, I just answered that question. So I hope that helps. So it was the deletion question. So that is really a popular question. And here's another one for deletion, but we've answered that as well. Okay, here's a question from Alan. Given the pandemic's impact toward events, are there alternative options being pursued to recoup the potential loss in funding? Tim, I'll let you answer that.
Well,
Repeat it, or you heard it?
no, I got it. Well, the answer is we need to raise as much money as we possibly can. And the way to go right now is crowdfunding. RedGive, Facebook, and the beauty of those two methods is they can be done from anywhere in the world by anyone.
Okay, thank you. Okay, Joe says, you mentioned one and done treatment, but CRISPR-based approaches are often incompletely penetrant, and won't a number of targeted cells remain unedited? Jan and Randy, one of you would like to answer that? I mean, I'll just start the ball rolling here by saying that there is plenty of data from mice models to suggest that even addressing 5–10% of the cells will result in improved symptom relief. So we don't need I mean, ideally we would target every cell, but we don't necessarily need to do that to get improvements. But Jan and Randy, do you have anything to add to that?
The only thing I would add too is you're right. I mean, it's unless we get to a situation where it's fully optimized and every single cell is targeted, you will have, you know, cells that don't receive the therapy. Right now, it doesn't look like you can redose. There's, for a number of reasons, there's immune reactions, there's different things, but science is always advancing, and these little hiccups could potentially be solved. And so it does leave the door open for additional treatments. And it's also possible that eventually we may have some kind of combination, you know, treatment as well, where you can utilize multiple therapies directed at different aspects of these mutations at the same time or sequentially. So I think right now it's still really early in what is possible, and that's actually a great place to be because it just leaves many possibilities as the technology advances.
Yeah, and I would just add that these are obvious problems that everyone in the field is aware of, and there's hundreds of millions of dollars being invested by NIH and companies to try to improve delivery to every cell in the brain and to promote and allow redosing and, you know, bypass these immune issues and to control the amount of protein expression from every gene that you deliver or every therapeutic. So these questions are being addressed by the general scientific community and are major focuses, and we're keeping a finger on the pulse. And as every advance happens, we'll try to put Rett syndrome at the front of the line for advancing these technologies into human trials.
Great, thank you. Okay, here's a question from Lena. She says, at one point there was a thought that protein replacement could be achieved by an external pump directly to the brain. Is this still in consideration? Also, would protein replacement be like gene therapy in terms of clinical trials? No phase 1, individualized to a point, etc. Randy, do you want to take that?
Well, there are approved proteins that are delivered into the spinal fluid by a pump for genetic disorders. And they have only modest penetration into the brain. So it is possible to do that. There is one that I'm aware of that is approved. It's a technology that people have tried. We don't believe that will be sufficient for Rett syndrome. It won't deliver uniformly to the brain, just to the surface. They won't deliver enough, we don't believe. So we're working on approaches that use transport mechanisms or other biologic systems to actively deliver the protein into the brain. And we're not the only group working on this. And one of the reasons it's not a major focus of funding for us is that there are many companies and many scientific groups that are focused on this. If anybody can solve it, we're paying close attention and we will leverage that. Dr. Shephardt has a novel, unique approach that we think has promise, which is at an early stage. And so we're working with her to see if we can actually make it be successful. So this would be, we think, really important, but today, even when you deliver by pump into the spinal fluid, you can't effectively deliver MECP2 to the brain.
Thanks, Randy. Okay, this is a question from Alina from Russia. She says, are biopharma companies going to take girls from different countries of the world for treatment trials? So this is a question I get asked a lot. So what will happen is when a trial is ready to begin, the protocol will be shared on clinicaltrial.gov website, which is a database of all clinical trials. And the protocol will list the clinics that are participating along with contact information. And then parents who are interested in learning more and potentially interested in enrolling can directly contact the clinics. And I don't think there's any criteria that says you have to live in the US, but that's not up to RSRT or maybe even the companies. It's really up to the clinicians at that point. Let's see. Here's a question from Alicia. What are your thoughts on pursuing gene therapy once available versus waiting for something like RNA editing or CRISPR? Well, CRISPR is a technology that's used in many of the strategies that we're pursuing. So the question really is thoughts on pursuing gene therapy versus waiting for something like RNA editing. That's a tough question, Alicia, and we all wish we had, a crystal ball. And I think that, that's a personal decision that probably has to do with the child's age and what kind of risk tolerance do parents have, and people may come to different conclusions. Some facts to share. So many of the strategies that we pursue today need to be delivered to the brain, and the gold standard for delivery to the brain right now is a vector called AAV9. And as of right now, you can only give AAV9 one time, because if you give it again, your body will mount an immune response to it. And so as of right now, if you've received one of these therapeutic strategies with AAV9, you wouldn't be able to get another strategy that also uses AAV9. Now, I do think that this is a problem that in time will get solved. There are a number of companies that are pursuing technologies that would, in effect, kind of make the AAV9 stealth so that it would go kind of under the immune radar screen of the body. And so in time, I do think that will be solved. But as of right now, that is the situation. Jan and Randy, do you have anything to add to that?
No, I completely agree. I think all of these challenges will be solved in the future.
Okay. Orietta from Italy says, you mentioned certain mutations being targeted for RNA editing. Can you anticipate a few? So for now, it looks like the mutations that lend themselves to RNA editing the best are the nonsense mutations. So like 255X, 270, 168. And in time, I think that portfolio of mutations that can be edited will grow. But I suspect to begin with, those are the ones that will be targeted. Do my colleagues have anything to add? Okay. Elizabeth asks, would these therapies require being operated on or tablets? I think she means like small molecule drugs. Many of these will be, they won't be tablets. They won't be pills. These will be therapeutics that are delivered, for example, intrathecally, like an epidural, similar, or perhaps other routes of administration, but not oral medications.
Yeah, I think the goal would be for these types of therapies to either be administered directly into the spinal fluid or to go IV into a vein in the body. Those are probably the delivery routes that are going to be most, that's what companies will be trying to do.
Okay. Jill says, as a Rett woman who's had treatment such as trofinetide, will gene therapy be okay later on when it becomes available? Yes. Having a small molecule drug should not impact being able to have one of these potentially curative strategies. Some people are asking about translations for the webinar. We will try to get translations to you. Please be patient, because we have to generate a transcript and then translate it. So don't expect it tomorrow, but we will try to get it to you as fast as we can. All right, here's another question from Alan. Randy will probably direct this to you. As an incubator, how does RSRT's engagement and investment evolve from the inception of a program to a pharmaceutical taking a therapeutic to trial or market?
Well, each time, each situation is unique. And so it may be, we may get involved when we find an academic who's funded by NIH, who's made a breakthrough discovery and has a large grant. And we provide animal models, cell lines, resources, collaborators, other thought leaders to help them take it forward. But we're very different from NIH. NIH asks a question, and in most funding groups is this an important scientific question? Is this scientifically interesting? Does it advance our scientific knowledge? Our approach is to ask the question, can we turn this into a therapeutic to treat children with Rett or individuals with Rett? So we're really focused on what would it take, because we have deep expertise or experience working in industry with venture investors or corporate leaders in public. We know the kind of questions they're going to ask before they will make an investment in a therapeutic program with a large amount of money. So we look at each program and say, what do we need to answer that would allow people, the leadership within that company, to make the investment in our program. And so it's unique every time. It may be answering a safety question, it may be answering an efficacy question, it may be answering a practical question. But the good news is that the foundation of research that we've supported and the network of investigators that we've recruited is just increasing all the time, and we can provide more resources every time. But they're the resources that are needed that will allow investors to feel comfortable funding development. That's our main focus every time. Did that answer your question? Or?
Well, we can't hear him, but
Do you want, Monica, Jana, do you want to add anything?
no, I think that's fine. I will just add a general comment that I certainly see a change in the interest level from investors and biopharma around Rett syndrome. That there is interest right now in these curative strategies, which a few years ago there was nothing. And so I think that's the most direct evidence of the impact that the type of research that we're funding is having. Without having funded these programs and de-risked some of these programs, companies would move on to other diseases. And now we're seeing them really, start to take an interest. There's 6 companies, there's more in the wings that we know of. And I think that will grow. And so for me, having been in this field now for 22 years, that to me is the most exciting thing. And these are MECP2 therapeutics, right? Not downstream targets, but things that are actually targeting the root cause of the disease. So
let me just add that I think Monica's vision, which is to develop resources that can be freely shared, that aren't encumbered by contracts. And everyone thinks that if an academic lab has developed a mouse model that anyone can use it. That's not the case. If they have a cell line that anybody can use it, that's not the case. That often it requires 6 months to a year of licensing negotiations, paying fees to be able to access those.
Especially for biopharma.
And that's just from academic center to academic center. Oftentimes they can't share it all with for-profit biotech companies and biopharma. And so the models that we've developed, the various animal models, the human cell lines that are critical to answer the question, does my therapeutic have potential to treat Rett syndrome? We've made freely available to biopharma companies and to investigators, and they're standard and they're high quality cell lines and animal models that are well characterized. And having them deposited at the animal models, for example, at Jackson Labs, who can actually do the studies. All the investigators, all the companies have to do is provide their therapeutic product. They don't have to, raise the animals, learn how to do the studies. They can be done there. We've made it really easy for people, for companies and others to answer the question, does my therapeutic have potential to work in Rett syndrome? And that's why we're seeing this avalanche and this flood. And it's an awareness of where the friction points are and where we can apply some grease to make things happen quickly. That's made this happen.
Great. Thank you, Randy. So we're at the hour mark now. So let me just say that we weren't able to get to all of the questions, but we can follow up via email with you to try to answer your questions. And if you have other questions after you've digested this information that come to mind, feel free to email me, monica@rsrt.org. We will have a recording. Give us a little bit of time, but we will have a recording available that you can share with anyone that maybe missed today, or that you can listen to again, and to help you digest. Thank you so much to all of you that are supporting us. Thank you. I hope that you will agree that we've put your donations and funding to good use, and that we will continue to do so. I wish all of you the very best. Please stay safe, stay well, and thank you for joining. Bye-bye.